Thank you everyone for coming. It's a huge privilege to be able to present my PhD thesis defense. Today I'll be talking about determining causal relationships between the microbiome and immune system. So when I came to graduate school, I knew for sure I wanted to study the microbiome and immune system, how they interacted with each other. I had absolutely no idea how I was going to do that, but I was convinced that it was the most interesting and exciting area of research. I can say for sure after working with Sonnenburg Lab for the past few years that opinion has just intensified, and I hope to communicate that passion, curiosity, and excitement with you all today. So before I start any project, I like to sit and think about the key questions so that the problems we're solving are both important and impactful. So the structure of my talk is going to be the key questions that we had at decision points to kind of drive the logical progression of analysis and experiments. So what are we studying? Why is it important? And how do we go about doing it? So starting with definitions, what is the microbiome? Microbiome is a set of microbes living on our skin and in our guts. This is what it looks like. There's a cross-sectional photo here. This confetti of gut microbes is separated from the rest of our body and our immune cells, which are shown here in blue, by this thick mucosal layer. It's kind of crazy to think about that everyone here has over 100 trillion microbes in their body, in their guts right now. The only thing that's separating them from the rest of your body is this mucus. This confetti of microbes is a very complex ecosystem, and it didn't get there overnight. Over 1 billion years of coevolution between mammals and microbes has led to a strong interdependency between these two systems. We rely on microbes just as much as microbes rely on us. So I showed you what the microbiome is and what it looks like. Why is it important? Well, as I alluded to, this interdependency has endowed the microbiome to be involved in nearly every organ system in our body. This diverse microbial community is responsible for, in particular, regulating metabolism in our immune system. So what do I mean by metabolism? Basically, this just means how our body harvests energy. So when we eat food, it's the responsibility of our stomachs and our microbes to digest it so we can harvest the energy from it. In particular, microbes digest fiber. For example, inulin is a fiber that our body can't digest on its own, so we rely on microbes to metabolize it for us. So when microbes come across a complex carbohydrate like inulin, it's able to break it down into short-chain fatty acids, which are small enough to cross through this mucosal layer, where they enter circulation and exert their effects on the rest of our body. So I mentioned at the beginning that microbes are physically separated from us in the gut. And the way that they're able to exert so many effects is by creating these small molecules that cross through and then enter circulation, affect systems such as the immune system. Obviously, our immune system is incredibly important. It keeps a state of balance in our body. We don't want to be too anti or pro-inflamed. If we have too much anti-inflammation, our body won't respond to pathogens and we can get infections. If it's too pro-inflammatory, we can develop chronic inflammation, and chronic inflammation leads to chronic disease. Inflammatory diseases are on the rise, such as inflammatory bowel disease, asthma, diabetes. These are all examples of diseases that continue to tick up year after year. Even scarier than this is the incidence of prediabetes, I should say prediabetes and obesity, which make up 37% and 40% of adults in the United States, respectively. These liminal states between health and disease that prediabetes and obesity exist in kind of show that there's a deteriorating health trajectory in our country, but also offer opportunities for us to intervene before more serious problems such as these diseases on the left here even have the chance to develop. So we're trying to push the needle on health. This leads us to the how, which is the most important question of my thesis and kind of the linchpin of my research career. How can we leverage the microbiome to better understand and treat inflammatory disease? In Sonnenburg Lab, we see the microbiome as the body's most powerful and important tool in defining the future of human health and disease. So the first, if we want to leverage the microbiome, we need to be able to study it. So how do we go about that? Typical workflow in biological research starts at the bench with mechanism in vitro. So we start with cell culture. We try to find a molecule that affects a cell type. Once we find these relationships, we validate them in mice or other animal models before finally going in to assess applicability in clinical trials in humans. So an example of how this can be perfect in a perfect world is Fleming's penicillin. Fleming found that penicillium in mold was able to inhibit bacterial growth in a petri dish. He then gave it to mice, saw that it stopped infection, finally gave it to wounded soldiers, and saw that again stopped infection in humans. So this is the gold standard for how this workflow can work perfectly. The reality of the situation is there's a huge translational gap between mice and humans. You can imagine we're very different from mice. And so for every one story of penicillin, there are thousands that don't translate. This leads to a valley of death in biological research. And when you consider two systems as complex as the microbiome and immune system, difficulties compound on each other and this gap gets wider and wider. So if we want to make meaningful changes in the near future at the microbiome immune axis, it really suggests that we need to change the way that we're looking at research as a whole. So as Justin alluded to, the innovation of my thesis is turning this process on its head and giving it a paradigm shift in how we study the microbiome immune axis by starting in human studies first, assessing applicability, how does the microbiome change, and then in turn, how does the immune system change? From there, once we establish these relationships, we can search for mechanism in vitro experiments, and then finally validate them in mice. Today, I'll be talking about the first two parts of this pipeline, human studies and in vitro experiments. In addition, I'll be talking about this really key chunk in the middle, that's the generating of testable hypotheses through data analysis. We want to parse all this data from the human studies and then be able to test all of the mechanisms in vitro experiments. So this is going to be my build slide. I'm going to keep coming back to it when I talk about different experiments and how they play into this experimental and computational pipeline. So the first thing I'm going to talk about is interventional human studies. Intervention is extremely important because it leads to causality. As Justin says, I'm very about causality. And so if we want to figure out what is causing what, like if X causes Z, as opposed to them just being associated with each other, we need to perturb the system and observe how it responds to change. If we want to change the microbiome and in turn change the immune system, how do we do that? There are a number of different ways that we can modulate the microbiome. Perhaps the most crude are fecal microbial transplants, or FMTs. This is my gut contents into your gut contents. This has been highly effective in things such as recurrent C. diff. We can also use diet to modulate the microbiome. I'll talk about that extensively today. And consortia of microbes or single microbes. These are often packaged as probiotic supplements. And then we'll kind of go on to what the future of modulating microbiome might be at the end of the talk. So focusing on the microbiome targeted interventions for today, I like to think about them in like a spectrum of specificity. So on the left side here, we have the less specific dietary interventions. We can modulate the microbiome through low fat, low carb diet, high fermented food, high fiber, ketogenic Mediterranean diets. We can do in the middle of specificity, which is swapping out one aspect of your diet, animal-based meat for plant-based meat examples. And then on the more specific side, we have supplements, those probiotics, which are the live microbes, or prebiotics, which are essentially fiber pills. So these are not just hypothetical examples. These are all studies that are being run by Sonnenburg and Gardner group. I've been extremely lucky to be able to at least a small part in all of these, but today I'll be focusing on the high fermented food, high fiber diet, as well as the probiotic supplement. So our key question here is, can a microbiota targeted intervention reproducibly change the microbiome and by extension, the immune system? Enter fermented fiber food dietary study, affectionately known FIFIFO. This was, as Justin said, published in Cell 2 weeks ago with Gabby Fragiadakis, and we're very stoked about it. So it's a 17-week longitudinal dietary intervention study. By longitudinal, we mean that we're tracking the same person and their change through time. So we have our baseline samples. This is before the intervention started. Ramp, where people slowly increase their intake of either high fiber or high fermented foods. Maintenance, where they kept that intake very high. And then choice, where people actually got to choose how much of the intervention they implemented into their daily life. So of course, I should mention that the high fiber and high fermented food diets were chosen from Gardner and Sonnenburg lab, seeing that they modulated the microbiome in previous work. So we looked at food logs. We also looked at the microbiome. Of course, we're able to do this in multi-omics ways. We can measure what microbes are there by sequencing the bacterial DNA in your stool. We can look at the products that microbes are making, their proteins or small molecules. This is all through patients' poop samples. As I alluded to before, your poop is a really good snapshot of what's going on in your gut. Okay, so what makes this study so special is that it is, other previous studies have shown dietary interventions affecting the microbiome. Maybe they'll do 1 or 2 measures of host health, but a lot of people didn't think it was possible to change your immune status with a simple dietary change. So what makes this study so special and so landmark in the field is it's the very first to implement extensive immune profiling in order to investigate the diet microbiome axis. Like I said, people didn't think you would see a change, but the vision of Sonnenburg and Gardner group was that the microbiome is powerful enough to make it happen. So when I say extensive immune profiling, we didn't want to just focus on 1 or 2 markers of inflammation and get ourselves into rabbit holes. We did over 230 immune parameters to assess immune status. We wanted a very comprehensive picture of how people's immune system looked so that we can get, not getting lost in the trees. We're looking at the forest is the way I like to think about it. Okay. So from here, we're collecting all this data. Even upstream from this, the first thing we need to ask ourselves in an intervention is it successful? Did participants actually change their diet to eat more high fiber or high fermented foods? So this is a real highlight of Gardner Group and Dahlia and Jennifer, who are key health educators in getting people to change what they eat. This is behaviorally very difficult, and they did swimmingly successful. Participants were successful in increasing their dietary intake of either high fiber or high fermented foods. So this is box plots. This green is the fiber intake. These are participants at baseline. So before the intervention started, they were eating around 20 grams of fiber a day. During the ramp, they slowly tick that up to a really high intake at maintenance, over 40 grams of fiber a day. And then for the fermented group, when they started, around 0 to 1 fermented food servings a day, and then ticked up to 6 servings of fermented foods a day. And for the fiber group, 40 grams of fiber. This is quite a lot. Very, very impressive on the dietitian's side. I do want to point out really quickly that in the choice period, this is where people got to choose how much they implemented that intervention into their everyday life. While it's not as high as the maintenance period of time, it was significantly higher than what they started at, and really suggests that they're going to make long-term lifestyle changes. Okay, so the dietary intervention was successful. Our next question was, does it change the microbiome? One of the measures to look at the microbiome is diversity. Did diversity change in the high fiber or high fermented food diet arms? So microbiome diversity is a little bit vague of a term, but in general, it refers to the number of unique species in your gut. So we sequence all the bacterial DNA. We say, how many of those are unique? How many do you have? In general, high microbiome diversity is good. It's associated with resilience and health. Low diversity is associated with vulnerability. People get sick more often, and people with obesity or chronic disease generally have a lower microbiome diversity. So the question again is, did microbiome diversity change? We saw microbiome diversity significantly increased in the high fermented food cohort, while the high fiber cohort had no change. So let's focus on the fermented foods here. You can see from baseline, they had around 100 microbes in their gut, and then this ticked up all the way into the choice period. So increases that were significant. What I really love about this plot is that it was most statistically significant in the choice period, which if you'll remember, they actually ate less than the maintenance period. So this suggests that it's not just about the number of fermented foods you eat on any given day. It's also about the consistency and the longevity of you doing it over time. In the high fiber arm, we did not see this change in microbiome diversity, which we were surprised by. Okay, so focusing on the fermented foods and how we saw this big change in microbiome diversity, was there a change in immune status that accompanied it? And so the big highlight of the paper, what everyone has been talking about, is that the high fermented food diet arm showed significantly decreased markers of inflammation. So I mentioned before, we measured 230 things. How many of those actually changed? Was it 1 or 2 things that decreased? No, we saw 19 different circulating inflammatory proteins decrease across intervention and 13 endogenous immune cell signaling parameters also showing marked decreases. This is what we're talking about when we say we're looking for trends. We're not picking out one or two things that happen to decrease. These are global immune status changes that suggest people are actually getting healthier and reducing their baseline inflammation after just a very simple lifestyle dietary change of eating more fermented foods. This is pretty wild. Okay, so we're going to shift a little bit from the fermented foods and go back to the high fiber diet. We didn't see any changes in the microbiome diversity. However, we were curious if anything changed in the microbiome. People are like, pounding fiber. Something had got to change in there. And so we looked at the microbiome functional capacity. And the reason we looked at this is I mentioned at the beginning, microbes are responsible for digesting things that we can't, one of them being fiber. Did they become better able to digest these complex carbohydrates? The answer was yes. As participants ate more fiber, their microbial capacity to digest it increased. So these are enzymes that microbes have that are able to digest plant-based fiber, and they all increased across time. So this suggests that there are changes in the microbiome that we're able to measure. People are better able to digest fiber. It suggests that if the intervention was longer, we might have seen more drastic changes. Okay, so we saw this change in high fiber microbial capacity. Did we see any change in immune status? Unlike the fermented food, which had global changes, the high fiber cohort actually had a personalized medicine or mixed immune response. So this is a heat map here. Every row is a different person and every bar is an immune parameter. So all 230 of those immune parameters are represented here. We have a number of different ways to monitor immune status. Inflammatory cytokines, endogenous signaling is how much immune cells are signaling, and then their capacity when they're hit with some type of trigger. So in general, broad strokes, if it's red, it means it's increasing, higher inflammation. If it's blue, it decreased, lower inflammation. So what you might see here is that it seems to be clustering. Some people seem to have a lot of reds, some people have a lot of blues and things like that. So what we did is cluster them using hierarchical clustering into 3 different groups. There were people who had significantly higher inflammation by the end of the intervention, people who were trending to be lower but didn't have any statistical significance, and then a low inflammation group 2, which did have a number of statistically significant decreases in inflammation. So from here, it suggests, we didn't see any signal in the entire group, but there were some people that did respond well to better to the fiber intervention. So this begs the question here, what determines if a person will respond to a high fiber diet with increased or decreased inflammation? And so what we saw is that the personalized immune response was dependent on baseline microbiome diversity. So the more microbes that people had in their gut at the start of the intervention, the better they responded to the fiber, the better their immune status at the end. So people that actually increased inflammation had low microbiome diversity, whereas people that started with high microbiome diversity actually decreased their inflammation. So you can see here is it's very complicated. Obviously, there's a lot of different things that are playing into it, but it suggests that our fermented food dietary intervention increased alpha diversity, microbiome diversity. If people eat fermented foods first and then eat fiber, it's possible that it could be a synergistic effect, and we could see even greater results than what we saw here today. Okay, so I'm going to wrap this up, wrap the Phi-FO study up and say it was the first and most comprehensive human study to profile both the human microbiome and immune system. On the high fiber side, we saw that the microbiome's capacity to digest fiber increased, and there was a personalized immune response based on baseline microbiome diversity. On the high fermented foods diet side, we saw cohort-wide increases in microbiome diversity and improved immune status defined by decreases in inflammatory markers. So I want to focus on the fermented foods diet a little bit, and we were curious what about the fermented foods was able to cause this huge change in immune status that we saw? What components were responsible for this? And so I think this begs the tangential question of what actually are fermented foods? So I Googled the definition of fermented foods, and according to the International Scientific Association for Probiotics and Prebiotics, fermented foods are those made through desired microbial growth and enzymatic conversions of food components. I don't really like this definition. I think it's kind of sterile, pun intended. So I decided to try and make my own, and I came up with something extremely imprecise. To me, fermented foods are the difference between spoiled milk and kefir. So this requires a story. If I was in front of you and I had a glass and I poured a fresh glass of milk and I had it in front of me, I set it out at room temperature, just on the counter for 3 days. After 3 days, I went and gave it to you and I said, here, drink this. It would smell bad. You'd probably turn your nose. Everything about it, visceral reaction would say, Hannah's trying to get me sick. Do not drink that. And you would be right. If, however, before I let it sit out at room temp for 3 days, I put a community of microbes that I knew would create a fermented food product, you would get kefir. And if you don't know, kefir is a fermented milk product. It's very similar to yogurt. And so it's a little effervescent. It wouldn't make you sick. You would be able to drink it. It would taste good. According to the fermented food study, not only would it not make you sick, but it might even make you healthier. So this difference between spoiled milk and kefir, I think, really captures the magic of what fermented foods are and how much we actually don't understand about them, which I hope to kind of break into a little bit today. So going back to that question, what components about the fermented foods make them decrease in inflammation? There are two that I want to focus on today. First being live microbes, that community of microbes that you put in the milk to begin with as it goes through fermentation process, and then the metabolites or the small molecules that those live microbes make across the fermentation process, both of which you ingest when you eat fermented foods. Okay, so I started with the interventional dietary study. From here, I'm going to grade into a probiotic supplement that encapsulates those live microbes, the potential component of fermented foods that made immune status improved in that first study. So can a probiotic supplement with strains also found in fermented foods confer the same effects as those found in the entire food product? The probiotic study is very similar. Went out to try and answer some of these questions, and it's very similar makeup to the fiber fermented study. It's an 18-week human interventional study. The big change between this one and the other is that the cohort of participants had metabolic syndrome. So for the fiber fermented study, those participants were generally healthy adults. These participants had metabolic syndrome. Very similar in the sense that we were looking at the microbiome through stool and the immune status through blood across time. So participants were selected based on their derangements for metabolic syndrome. According to the International Diabetes Federation, you have metabolic syndrome if you have a waist circumference above the threshold here. It's defined as central obesity, and you also must have 2 or more of the other inclusion criteria above these guidelines. So to give a quick snapshot of what our cohort looked like, each of these rows in a heat map here is a participant. It's a red block if they're over that guideline and a blue if they're under. And so because it needs to be 2 or more, we added them up in this final column here, and you can just see kind of the spectrum of metabolic syndrome that people were on during this study. For the probiotic supplement itself, we determined what bacteria were in there using 16S sequencing. It was about half bifidobacterium and lactobacillus, both of which are found in fermented foods. However, this cocktail of 3 different species were chosen because they were all previously reported to be associated with success in small clinical studies focused on metabolic effects. Okay, so the primary outcome for this study is, can this probiotic supplement improve parameters for metabolic syndrome? The short answer is that it did not have treatment arm-wide effects in improvement of our main driver, metabolic syndrome participants. However, we did show, similar to the fiber group, a collection of responders and non-responders, people who seemed to improve and people who did not. Again, this was determined by hierarchical clustering. People who responded had significantly decreased levels of triglycerides and diastolic blood pressure. So again, we're seeing this kind of personalized response. Some people respond, some people don't. I don't have enough time to talk about all the different parameters and microbiome that we tested in this study, but I will go into the main question that I had: what differentiated the probiotic responders and non-responders? The main covariate that we found was diet, actually. Specifically, people that responded better to the probiotic supplement had higher intake of total and added sugars. This was very surprising to us. Sugar is not something that we usually associate with health. We have a few hypotheses on why this might be. Maybe that it was, stabilizing the microbes in the small intestine or something like that. But honestly, it just points to how much research needs to go into studying the intersection between diet, supplements, and microbiome. So I'm going to end this study here, and hopefully it'll be submitted and reviewed soon, so you can see all the really cool data that we have tracking the microbiome. But human intervention, mixed response to probiotic supplement, diet seems to determine how you respond to it. Okay, so I'm going to wrap up the interventional human studies there, go to a different flavor of human studies and with the cross-sectional. So from the interventional studies, we're able to see some microbial immune associations. I'll talk about this a little bit more later, but now we wanted to combine both of these studies and get a larger feature set in a cross-sectional study. So cross-sectional means instead of looking at people through time, we just look at them at one time point. And so what we did is took the baseline of the fiber fermented study, which were healthy adults, and the baseline of the probiotic study, which was metabolic syndrome. And from here, you can tell we have a healthy versus metabolic syndrome sample set. So healthy versus disease. I think this is a really, really cool data set. You can look at parameters of metabolic syndrome, microbiome, functional capacity of the microbiome, their immune status, metabolomics. It's just this really rich data set. It's really hard to get high quality human samples. And we'd be missing something if we didn't take this opportunity to kind of combine them. Today, I'll be talking about the metabolomics profiles of these healthy versus disease cohorts. The reason I'm focusing on this today is that if you remember at the beginning of my presentation, I said that microbes produce metabolites that enter circulation and are able to exert effects on our bodies. So these small molecules are extremely important in modulating inflammatory signaling, and I'm curious how they differentiate between healthy and disease. Put another way, are there metabolite signatures indicative of states of health and metabolic syndrome? So the first thing to do with this is define what we're saying is healthy and what is metabolic syndrome. Very simple, same from what I did before. Following the International Diabetes Federation guidelines, healthy defined as less than or 1 parameter above threshold, intermediate is equal to 2, metabolic syndrome is 3 or more. So here, exact same heat map, just now with 79 participants. Red is if they're over the threshold, blue is if they're under. And here you can see we have a nice gradient of what we're calling healthy and disease. Okay, so from here, this is our breakdown of the metabolic disease states. 27 were healthy, and then 20 apiece for both intermediate and metabolic syndrome. So from here, we wanted to see what the small molecules were in their stool and in their blood using a high throughput liquid chromatography mass spectrometry pipeline developed by Schwo and Will and Sonnenburg lab. So we took 70 stool samples of participants and 74 serum samples and ran them through this high throughput metabolomics pipeline. From a spiritual level, if you want to know what it took for me to get 70 of these stool samples through this machine, please ask me about my poop catastrophe later. Okay, so from here, we look at all of the molecules that are in our stool and serum samples. Are there differences between our metabolic disease states? Can metabolites in the stool differentiate if you're healthy or if you have disease? We found 400 different metabolites that were detected and annotated in the metabolomics pipeline. We did not see a differentiation between disease states. So this is a peak away plot. There are 400 metabolites. I can't show all of them on one plot, but what we did is squish them into 2 axes. The only thing you need to know about these plots is that each dot is a person and their color corresponds to the metabolic disease state. The closer the points are to each other, the more similar they are. So you can see here that there's no real, there's sprinkles. Everyone's kind of mixed together. There's no differentiating factor between them. On the flip side, we looked at the serum metabolites and we were curious if that differentiated health versus disease. We found that they did actually significantly determine whether or not someone was healthy or diseased. So this is what we would expect when there is a signal. All of the red dots are kind of clustering together. All the blue dots are kind of clustering together. And then the yellow, which is intermediate, is actually kind of a smear between the two, which I thought was really cool. And then this is statistically significant with this p-value here. So it's kind of unsurprising that the serum metabolomics profile was different between the two, given that metabolic syndrome is measured through a lot of blood samples, cholesterol, triglycerides, things like that. But it is really cool proof of concept that the metabolomics pipeline is very powerful and leads to validation of what we would expect. So this looks at all of the metabolites that we were able to detect. Are there specific ones that we're able to point at and say differentiate the two groups from each other? We found 16 serum metabolites that were significantly different between the two groups, healthy and metabolic syndrome. Some of them were increased in the metabolic syndrome cohort, suggesting that it might be inflammatory or associated with more inflammation. And then some of them were higher in the healthy cohort. Interestingly, the intermediate, again, was kind of in between the two, suggesting that there's a spectrum of health and we're able to track it through your serum metabolites. Okay, so from here, we looked at cross-sectional data and we found disease-associated metabolites. From here, I'm going to go into our in vitro experiments and the hunt for causality. Okay. First in vitro experiment is metabolomics again, a deep dive into fermented foods. So I talked about there are two components of fermented foods that we were interested in seeing if they had an effect on the immune system. First was the cohort of microbes, which we investigated a little bit with the probiotic supplement. Now we want to look at the metabolites that are in those fermented foods. What does the metabolite profile of different fermented foods look like? So instead of doing poop and blood through this machine, we did a whole bunch of different fermented foods. All of this work is with Sean Spencer and Eliza Caffrey. What we did is ran the metabolomics. This is a heat map showing what was in there. I'm not going to go into like specific metabolites that were in there. Darker blue means they were high, lighter blue means they were low. We're just looking at trends. And again, we're seeing that the fermented foods cluster by expected type. So all of our dairy ferments, our two types of yogurt and our kefir are on the left here, and all of our vegetable ferments are on the right. And so this metabolomics profile is just showing that it classifies fermented foods very well. I want to focus on kefir because it was my example of what makes fermented food so magical. So focusing on dairy ferments, in particular kefir, let's start at the first part, milk. Is the metabolomics profile able to differentiate milk based on source? So we had 3 different milks here: cow milk, human breast milk, goat milk. We're able to predict with 100% accuracy based on 15 metabolites where the milk came from. From here, we're interested in whether the metabolite profile of milk pre and post fermentation differed based on source. So before we're looking at where did the milk come from, now we're looking at milk and kefir. What we saw with the metabolite profile, again, things that are close together mean that they're similar to each other. The milks are all on the bottom. So this is goat milk, cow milk, and human breast milk. They're all on the bottom, differentiate against pico-A. When we looked at cow kefir and goat kefir, they move up pico-A2, so suggesting that source differentiates on one axis and fermentation state differentiates on axis 2. Again, distinct metabolite patterns that we're able to detect. Okay, so I talked about milk and kefir. The next question is, can we track that fermentation process? Can we see what happens during that magical period of time that milk becomes kefir through their metabolites? The only way to do this is to do a home ferment. This presentation wouldn't be complete without a home fermentation. So this is my apartment. This is my counter. We took goat milk, put it in this beaker here. Like I said before, we just put a community of microbes. This is Justin and Erica's kefir grains, plopped it in there. This is kefir grains. They're a really sticky mixture of bacteria, yeast, bacterial cell coat. It's pretty gross, actually. We plopped that into the milk, and I just set it on my counter for 3 days and collected collected samples at all of these different time points. 3 hours every 2 hours. It really messed with my sleep schedule, but it was worth it because the data is really beautiful. We can see metabolomics over time tracks with the process of goat milk to kefir. So what I want to share with this is that earlier time points with milk are really spread out. There's no homeostasis early on in the fermentation process. It suggests that the microbial community hasn't really reached a stable level. However, once the fermentation process continues, the points get really tight together, suggesting that it's becoming stable and the community is not really changing very much. And we can look at this through their metabolites. So again, this is looking at the entire metabolite profile. are there specific metabolites that we can track that are microbially produced and consumed over the course of fermentation. So we ran a random forest and it pulled out 14 metabolites that are able to predict fermentation hour with 95% accuracy. If you tell me how much of each of these 14 metabolites is in, I can say what hour of fermentation it's in. I think that's really, really cool. I also love these plots. It's a little overwhelming, but I love these plots because it just shows how beautiful like biology and math intersect with each other. These are really great plots showing hippuric acid just getting consumed by microbes, mevalanoacetone just increasing, and we can just track all of this. So I really like this plot. I wanted to look at it all on one scale though. And so what we did here is modeled each of these 14 metabolites over time using a logistic regression. So this is fermentation time in hours, the amount of metabolites scaled, just so I can see them all in one plot. All of these purple ones are being produced over time. All of these brown ones are being consumed. And so from here, I knew that there are more than 14 metabolites being produced and consumed. So I wanted to expand this feature set, see what else is out there. So what we did is did a Spearman correlation with each of these 14 metabolites and every other metabolite that we were able to detect in the kefir, and found almost over 180 different metabolites that are microbially derived, at least in the sense that they decreased over time or increased over fermentation time. So this gives us an expanded feature set of microbial derived metabolites during fermentation. So from here, this is a lot of lines. I wanted to pick out, something that we would expect. I wanted to give my penicillin gold standard. Classic fermentation takes lactose to lactic acid. So it's, we started with milk. It has lactose in it. If you're lactose intolerant, that's why you're sick. Microbes are able to metabolize this into lactic acid. So from these lines here, I can track lactic acid being produced over time, and then a really nice curve showing lactose being consumed. In addition to that, we know there are a few intermediates between these two steps, and we can also track those through time with some different kinetics. So this is just, very preliminary data. It's not ready for prime time, but I just really like the way that we can track fermentation, microbial communities over time, and it's really powerful and potentially could be used to leverage time course metabolomics data to construct pathway maps of metabolism for small molecules that are upstream and downstream from each other, much more complicated than lactose to lactic acid, and potentially uncover really novel pathways. I just really like this plot because it encapsulates everything that I've done in grad school. I think there's longitudinal sampling, there's fermented foods, microbes, fermentation, statistical modeling. If I didn't have all of that experience, I wouldn't have been able to make this plot. Okay, so that was our first in vitro experiment. The last thing I'm going to talk about today is how do we actually take these metabolites and find those that are causal for immune modulation. So we take, all of these human studies that we did, the data analysis that suggests microbial immune associations, disease-associated metabolites that are produced over time during fermentation. Which of these are actually causing changes to the immune system, that causality? Okay, so the first question is, how do we even test this? How do we test immune modulation? First is, we just did human blood samples. So we take a sample of someone's blood, we expand their immune cells, or PBMCs, they're just immune cells, and then, we look at how much inflammatory signaling. So when your cells are just not doing anything, they're not being poked, they don't really signal with much inflammation. However, when they come into contact with a bacterial cell wall component, they get stimulated and they start producing inflammation. This is normal, that should happen. This is why we don't get infection every time you come into contact with a microbe. The innovation here is looking at priming immune cells before they're stimulated to see how that modulates their inflammatory signaling. So in this step, prior to being stimulated with bacterial cell wall component, we bathe them in metabolites and see how that changes how they respond to bacteria. So for example, if we bathe the cells with a metabolite that's not immunomodulatory, we'd see no change as concentration increased of that metabolite. So there's no change. This is a negative result. On the flip side, if the metabolite has immune modulation properties, we would expect to see less inflammation the more metabolite we incubate it with. So if you don't understand anything I just said, the only thing you need to know is that this means that the metabolite does not have immune modulation effects. This means it does. Okay, so from here, we're going to go all the way from the back to the front. Interventional, we're going to start with the high fiber or high fermented food diet, how we looked at microbial immune associations and then directly tested those in vitro experiments. So one part of the high fermented food cohort that we actually had to put in supplement, but is really cool. We did serum metabolomics of these participants, looked at how they changed over time and how they associated with the inflammatory change. And so this is small molecules or metabolites that we found in their blood. These are inflammatory cytokines and the statistically significant associations between the two. So this is kind of getting at some of those microbiome immune axis that we're so interested in. And so the most significant was actually the set of bile acids. So we're interested, are these bile acids actually modulating the immune system or are they just associated with changes? Are these bile acids associative or are they causal for immune responses that we can measure in vitro at the bench? Okay, so this is the same experimental setup. We have human immune cells. We stimulate them with LPS after they've been bathed in all of these different bile acids. Lithocolic acid is a bile acid that's already been shown to be anti-inflammatory, so decrease inflammation. And in our hands, we saw the exact same thing. As you add LCA, the amount of IL-6 or inflammatory protein decreased. For the other bile acids, cholic acid, glycolic acid, we didn't see any change, so we don't think they're immunomodulatory. However, taurocholic acid has this really interesting trend here where it seems to be decrease IL-6 production at a certain concentration, but then it can be too much and then it actually becomes, it goes back up to normal. So I'm really excited about this pipeline. I think it shows in our hands that we can get it to work. It's only a matter of time before we churn through the other metabolites that we talked about and that we made hypothesis generation on for us to find microbial derived immune modulatory elements. So this is all of the body of work I'm going to talk about today. If I could say in one sentence, all of my graduate work, I would say that I developed an experimental and computational pipeline for identifying immunomodulatory metabolites. All of this was grounded and anchored in that first important question: how can we leverage the microbiome to better understand and treat inflammatory disease? The more I learn about the microbiome, the more I'm convinced I am that it is the future of human health and medicine. I don't think it's outside of, I think that we can expect within our lifetimes that microbiome is able to be used for precision medicine to determine who responds to cancer immunotherapy and not, treatment of autoimmune diseases, vaccination efficacy. On the population-wide, I think microbiome can be used to decrease inflammatory status, slow or reverse the global spread of chronic diseases. This is the most exciting field of research to be in. The next few years are going to define its trajectory, and overall, I just feel really lucky that I was able to study it here at Stanford. Thank you. I'm going to go into acknowledgements, straight into acknowledgements in case people have to jet out. I worked with so many people in Sonnenburg Lab and beyond. I want to thank so many different people, which I will in my acknowledgements. First, I want to thank my committee. I wrote this out because I didn't want to mess up my acknowledgements. Thank you for being the absolute dream team committee that I could have ever imagined. Every aspect of my thesis was built and inspired from your research and scientific vision. I also want to thank the Gardner Lab. I worked the most with Christopher, Dahlia, and Jennifer, who are absolute key health educators, dietitians. I learned so much about food and nutrition from you guys. Thank you so much. Okay, Justin and Erica, I found this really nice picture of you guys on the internet, and then I looked through my own camera roll trying to find some pictures that I might have of you. Justin, this is from 2018, so it's pretty old. You have to remind me what what it is that you're chugging on the ground on one knee. I think it was a Smirnoff Ice, but unless Eric is asking, then it was just a bottle of water. So that was from 2018. This is from 2019, where Justin decided to wear a different sweater, but the same dad vibes. This is him teaching his daughters how to aim with a Nerf gun. So a lot of mentorship, really important skills. In seriousness, Justin, joining the Sonnenburg lab was unsurprisingly the best decision I made at Stanford. The scientific vision, mechanistic insight, leadership, and mentorship you've shown me is absolutely incredible. I am greatly inspired not only by the way you approach problems, but also the deep grounding you keep for prioritizing connection and friendship. The lab is a direct reflection of the culture you value, and I'm endlessly grateful for being a small part of it. Thank you for believing in me, encouraging me to grow at every turn, and pushing me to be a better scientist, communicated as a compliment sandwich, of course. Erica, joining Sonnenburg Lab was unsurprisingly the best decision I made at Stanford. The insight you have in identifying problems and the clearest communication thereof is nothing short of transcendence. I have learned so much from you. Thank you. To my lab family, Gabby, I will never be able to thank you enough for being my first advocate and inspiration at Stanford. I truly cannot imagine in any capacity what my experience in grad school would have been like if you had not taken a leap of faith and taken me on as your first mentee. Thank you for teaching me how to code, just how to code, but also with patience and statistical rigor. Thank you for giving me the confidence to dive headfirst into a new field. Thank you for being my mentor. Kat, you are one of the best and most conscientious scientists I know. More important than that, you are one of the very best humans I know. Thank you for showing me what it means to be upstanding and kind in every endeavor. Rebecca and Dylan, I couldn't have asked for better people to join the lab with. I'm so glad we backed Justin into a quarter and convinced him to take all of us on. I value my friendship with the both of you very, very dearly. Will, thank you for inviting me to embark on the most exciting adventure into the world of biotech. I don't know where it's going to take us, but I do know we will pour absolutely everything we have into it. Kelly, thank you for being one of my closest friends in lab and all of those endless dinners at Sweetgreen and Mendocino farms. Brian, in this absolute brick house of an apple pie, you have gotten me out of too many tight spaces to count. I hope to emulate even a fraction of the incredible patience and kindness you have shown myself and everyone in lab over the years. Sean, I have never met anyone with your brand of spunk and curiosity. I will never have the deep, unbridled love for fermented foods that you and Eliza have, but one can dream. Michelle, you are my forever favorite lab manager. Your friendship and wit are one of a kind, and I feel very lucky to have you as a dear friend. To everyone in lab, I just felt like I was in the best innovation ecosystem of all time. I think this picture really encapsulates the spirit of the lab. I think from this question, people could ask, do you guys even ever work? And it sounds cheesy, but when it feels like play, it doesn't feel like you're ever working when you love what you're doing. To my mentees, Corey, Evelyn, and Miles from SSRP, and Eliza, my rotation student, you're also incredibly bright, warm, and talented. I know you all will go on to do amazing things in your scientific career and beyond. I feel lucky to have been a very small part of that journey. Eliza, I know you're going to go and take on all of the fermented food projects into heights that I can't even imagine, and I can't wait. For all of you guys, thank you for the privilege of mentoring you. To my mentors, Penn State, Dr. Robertson, thank you for taking me on and letting me join your lab as a high school student. I really learned to love research in Robertson Lab. To Greg, I'm a scientist because of you. I truly cannot emphasize enough how much of an impact you made on my life and professional trajectory. Despite all of your persistent urgings to never go to grad school, I'm sorry, I just did not listen to that one piece of advice you had. 90% of the scientist I am today is because of your mentorship and trust in me from the very beginning. Thank you for everything. I hope to make you proud. Catherine, thank you for teaching me to be a biochemist. That obsession with causality and mechanism is from you. I hope it's clear from my talk today how much of an impact you've made on me as a scientist. Mar, you showed me what it means to be a boss in Silicon Valley. In addition to being a steadfast and dedicated mentor, you believed in me before I believed in myself. I can't thank you enough for the amount of inspiration that I get just from being around you. To all my beautiful friends, Erica and Abigail, my OG best friends who I consider my little sisters, Hannah and Therese, who I consider my older sisters, Elaine and Erica, my Twilight crew, the best roommates ever, Sarah and Alex, my sassiest friends here. Erica Schwartz, you are undoubtedly the coolest person I know. Thank you for being one of my best friends and sharing a deep, genuine love for storytelling. You showed and gently guided me to what it means to be a creative and artist. Nicole, I wouldn't have wanted to move from Delaware and become California girls with anyone else. Thank you for the steadfast friendship and laughter over the past 8 years. Rosa, you are my muse. I cannot express how much you inspire me every single day to be the best human that I can be. You're the most intelligent, thoughtful, and kind person I have ever met. I feel incredibly lucky to have you in my life. So much of who I am and who I strive to be is because of the impact you've had on my life. Thank you for being you. To my family, so many beautiful people in my family. To my Aunt Nancy, thank you for fostering a love for reading from the very beginning. Not a year went by that you didn't gift me a book or something to encourage my creativity. To my grandma Stella, think about you every day. I love you. Finally, to my parents. This picture is them in China. I'm here picking me up from the orphanage. When I think about what had to happen to go from this picture to where we are today, I get freaked out, honestly, because it seems like such an impossible thing to happen. I still don't know how any of this happened, but I do know that it's only possible because of the love, dedication, and care you have showered me with my entire life. I cannot thank you enough for everything you've done to support me through everything. You both continue to surprise me with your capacity to love every day. I can't encapsulate how I feel about you in one PowerPoint slide, but this is my shouting off the mountaintops that I love you two so very, very much. Thank you to everyone for being here. I really appreciate it, and I'll open the floor to questions.
All right, the floor is open to questions. Maybe our panel will come back on, we'll see them live. Maybe Susan can't. Oh, the chat's pouring in. Hannah, I hope you're watching the chat. Yep, take a big breath there. That was amazing. I don't know, I was off camera crying, so I don't know how you did that without tears spilling out of your eyes, but good job. I actually have a power issue. I'm going to close my video and mute for a minute while I switch power sources. I'll be back in a minute.
Great job, Hannah. That was amazing. Michael, do questions normally come in through the chat, or are people unmuted as they raise their hand?
Some of both. I'm seeing a consistent theme in the chat. People seem to be pleased with the quality of the presentation, as am I. Somebody mentioned Mario Kart. I think we're gonna have to follow up on that in the private session.
Looking forward to it.
There's a question in question and answer from Tyler.
Yeah, I can read that, Susan, since you're on a slow connection.
Okay.
It says, great talk, Hannah. In the high fiber or fermented food diets, did you identify any superfoods that are the ideal foods to consume in terms of content, patient compliance, accessibility, etc.?
Yeah, that's a great question, Tyler. We did look at if foods were directly associated with changes in microbiome diversity. So yogurt and sauerkraut brine, or like fermented foods brine, seem to have the strongest correlation with microbiome diversity. I think it's hard because people got to implement the intervention in whatever way fit their lifestyle. So instead of everyone eating the same thing, people were making their own kombucha, eating their own yogurts and things like that. So it's hard to say what was the best because everyone did something different, but those seem to have the biggest effects, probably because people were just able to eat the most of them. I think a next area of research is making fermented foods that are enriched in the causal components for immune modulation, so that instead of eating 6 cups of yogurt, people can eat 1 or 2 and have the same effects.
Great. And another question came in from Ed McCarsky. Indeed, very clear. What role does gut permeability play in your observations, particularly when looking at patients with metabolic syndrome versus normal?
Yeah. Oh, gut permeability is hard because there's not like a perfect measure to see how leaky someone's gut is. It's hard to compare also the fermented study versus the probiotic study because we don't know if the fermented foods would have the same effect in people with metabolic syndrome. So it would be interesting to have a future study that looked at gut permeability as another parameter and seeing if it changed over time or if it determined if people responded to fermented foods in different ways. But yeah, that's a great question.
There are other questions popping up, but Ed had a quick follow-up. What about the amount of bacterial DNA in serum?
Oh, we didn't measure that actually. That's something that we could look at in the future. That's an interesting question.
And then Nina Horowitz, amazing talk, Hannah. Did you see any differential effects by age, sex, or amount of exercise?
We did look at that in both the fiber personalized response as well as the probiotic personalized response, and there were no differences in age, sex, amount of exercise, weight, any of that stuff. Yeah.
Sean Spencer asks, from your fiber and probiotic intervention, it appeared the most responsive individuals were less diseased, inflamed at baseline. How do you interpret this observation? Is a less diverse microbiome in an inflamed host more difficult to change?
Yeah, that's an awesome question. Oh, that's a really hard question to answer. I mean, people have states of the microbiome for sure. I mean, we showed in one of the papers that when people lost a ton of weight with a different diet, their microbiome just kept pulling them back to the same state. So probably some type of really large overhaul is going to be needed to get people to get out of a particular microbiome that they have established. That's why FMTs seem to be so effective in some instances. So more difficult to change. Yes, I would definitely say that's a.
And Christopher, I took over your chair responsibilities while you were changing power. Go ahead and pick it up.
No, this is fine. I'm happy to have you read the questions, but I can take over. So it looks like we're at Gabby's question. With all the permutations of patient cohorts and dietary interventions, what would be your next dream dietary intervention study?
I think either the synergistic effect of the fermented foods and fiber, like giving people fermented foods, seeing their microbiome increase, and then seeing if we can push the envelope even further with fiber. Other than that, I would love to play around with fermented foods and try to get kind of what the first question was, is there a perfect fermented food? I'd really like to see if we could get something like that.
Okay. And Brian Merrill asks, amazing, Hannah, given the personalized immune response you observed following increased fiber consumption, dependent on original microbiome diversity, do you think increasing that diversity? Oh, here, it's the same question you just had, by consuming fermented foods first. Sorry, I should have read the whole question before
Brian,
I started.
that's my dream study. You just answered the question.
There we go. Compounding effect on reducing inflammatory markers.
Yeah.
Okay. Ed is back. Wait a sec, he came. From Jason, we have a question. Do these FIFO results track across cultures with more or less fermented foods? Or conversely, are there cultures with more fermented foods that have differential metabolic disease prevalences?
Yeah, I mean, the metabolic syndrome and diseases are highest in industrialized countries like the United States. So, I mean, it would be, I don't know the answer to like whether, Korea's intake of kimchi is way higher than anywhere else in the world, if they have lower rates of metabolic syndrome and things like that. That's an interesting question. we don't know how they track across cultures or different cohorts of people since this was just the first one. But I like these questions because I think it's getting people like, oh, you should test this, you should test that. I mean, the space for more interventional human studies is wide open.
All right, Ed's got another follow-up. Caspase-8 is a wonderful pro-death mediator, but not particularly known as a cytokine, yet clearly changed dramatically in a couple of settings. What does this mean? Is caspase-8 being released from cells?
Ooh. Caspase-8, was that one of the ones that was increased in response to the bile acids? I don't know the answer to that question. I think we were looking for, again, the trends of lots of things changing. So something that people were saying is, oh, IL-10 decreased, and that's supposed to be associated with lower inflammation. I think with the feedback loops of everything feeding into everything else, seeing global decreases is kind of what our narrative was, as opposed to one thing changing cell-mediated death and things like that. But that's a great question, Ed.
And Ed, if you have any theories, we'd love to hear more about them. I saw a question fly by just asking Hannah if any of the studies in your data analysis has made you change your diet.
Yes, I definitely eat a lot of fermented foods. I tell everyone that I know to eat fermented foods. My mom likes kefir. Yeah, I would say, I mean, people should still eat fiber. I don't want people to go home with that story. So eat both fiber and fermented foods. That's what I do. Yeah.
So I have a practical question for you, Hannah. Can you help describe to those who are listening 6 servings of fermented foods a day? That must be all of your calories per day. Do you want to help put that in perspective, what 6 servings a day was?
Oh, well, it's definitely not all your calories. I mean, people, if you drink like sauerkraut brine, you don't have to actually drink that much of it to get 6 servings a day. Kombucha is like a thing of kombucha is 2 servings, a thing of yogurt is 1. So, 2 things of fermented foods per meal. I mean, not impossible. It's not that many calories. Fermented foods aren't calorie dense, I would say.
Right. Yeah, it's really, from a nutrition perspective, half a cup of yogurt or half a cup of sauerkraut is a serving. So if you had a cup, that would be 2 servings right there. A normal bottle of kombucha is 2 servings in the store.
Yeah. And some people were saying that it's too much salt, but you don't have to eat salty fermented foods. You could also have yogurt.
Yeah. Okay, questions are slowing down. We could take some from the committee or...
I hate to be a downer, but I think we're gonna have to go to the private session.
Okay, got it. Well, good. A lot of people had a chance to get some questions in, and Hannah, you did a great job there. So, Michael, thanks for keeping us on track.
Totally. Yeah, thanks everybody for showing up. This was awesome, and huge congratulations to Hannah.
Yep.
Thank you, everyone.
Bye to the fan club. They're signing off. So, all right, now I actually don't know how this switches up. Do we get off and join another link?
Yes, the link that
Okay.
Deborah sent, there should be a separate one for just the committee.
Okay, I'll go find that. Thanks.
Okay, yeah.